Sir, I can't see the recorded class. They ask for SSS.
Okay, good morning everyone. Thank you very much for joining in the class. For recorded classes, I already approved your access. So, okay. So, if anyone yet to get access, just email me. I'll try to solve it. Okay. Is
Okay,
it
sir.
clear? Okay, thank you. Are you seeing the screen now?
Yes, sir.
Yes, sir.
Okay, so today we'll discuss about plasma membrane. If you have idea about plasma membrane, right? So,
Yes,
what is
sir.
plasma membrane?
Outer layer of a cell.
Okay. So, it is composed of carbohydrate, protein, or lipid.
Protein and lipids.
Protein and lipids.
Okay. Is there any carbohydrate here?
Um, a carbohydrate could be there maybe in the form of a conjugated protein, for example, like glycoproteins, which could be, I don't remember exactly, so maybe like there as channels for transporting some substances in and out of it.
Okay, great. Okay, so plasma membrane are actually outer layer of a cell, right?
Yes, sir.
Okay. So it mainly contains lipids and protein. Am I right?
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay. And plasma membrane is also called cell membrane, isn't it?
Yes, sir.
Yes, sir.
Okay. So it's composed of phospholipid, protein mainly. Okay. So why it is called like phospholipid bilayer?
Because there are two layers of the phospholipids who are facing opposite to each other.
Okay, great. So bi means two, right? As there
Yes,
is
sir.
two layers, that's why we called it bilayer. Okay. So phospholipid composed of two types of things, right? One is head and another is tail. Okay. So the head is hydrophilic and the tail is hydrophobic. So what does the meaning of hydrophilic?
It attracts water, and hydrophobic is where it fears water, so it doesn't go close to water.
Okay, we all know that the word feel means love, right? So hydrophilic means water loving. Okay.
That's it.
And phobia means like afraid, okay, anxious, like that. So this tail anxious about water. Is it clear?
Yes, sir.
Yes, sir.
Okay. So another thing we can see from the figure is protein, right?
Yes,
So
sir.
two types of protein we are seeing here. First one, integral protein, right?
Yes, sir.
And another is peripheral. So why its name integral? Anyone?
It's
Cause it's connected to
situated
both
into
layers.
the layer.
It's in between two layers.
Yes, it's in between two layers. So it's integrated in the lipid bilayers. Okay. And why it is called peripheral?
Because it's in surface.
Sorry.
Because it is in the surface.
Okay, it is in the
Layer.
surface. Okay. It is just in the one layer. Okay. Is it clear to all?
Yes, sir.
Yes,
Okay.
sir.
Another thing we can see here is cholesterol, right?
Yes, sir.
Okay. So we'll discuss in detail about each now gradually. Okay. Step by step. Okay. So here is the plasma membrane component. So we already discussed about phospholipid, cholesterol, integral protein, peripheral protein, right? But
Yes, sir.
we didn't talk about carbohydrates. So how carbohydrates present in the plasma membrane? They actually are in conjugated form. They actually are conjugated with proteins or lipids. Is it clear?
Yes, sir.
Yes, sir.
Okay. So in combination, they are both called glycocalyx. Okay. So glycocalyx means glycoprotein and glycolipid. Is it clear?
Yes, sir.
Okay. So what this line actually indicate? What these lines are indicating?
Sir, cytoskeleton filaments.
Okay, cytoskeleton. So they are like somehow this membrane is connected with cytoskeleton, isn't it?
Yes, sir.
Okay. So this is an integral protein, right?
Yes. Sir.
So and here is label protein channel. So why it's named channel? It's a kind of integral protein as well. So why it named as channel?
Because something passes through it.
Okay. Something can pass through or pass out. Okay.
Sir, then what is the use of the integral membrane protein here then? If it's not working as a channel?
Okay, integral membrane protein, there are many types. Okay. We'll
Yes,
discuss
sir.
it gradually. Okay. So
Okay.
it's just an example. Okay. That
Okay,
it
sir.
may act as channel. Okay.
Understood, sir. Okay.
So any question from here?
Sir, about the glycocalyx, like what are their functions? Like what are the function of like glycoprotein and glycolipid? Are they acting like as protective surfaces or what are they acting as? Sir?
Okay, great.
What
Okay.
is
So here you can get idea about this function of these carbohydrates. Okay. So from the picture, we can see that these carbohydrates are in conjugation with either protein or lipids. Okay. So these
yes, sir.
carbohydrates are actually 2 to 60 monosaccharide units and they may be straight chain or branch. Okay. If
Okay,
we
sir.
go back to the figure, they may be either straight chain or they may be either branch. Is it clear?
Yes, sir.
Okay. So what is the function of this carbohydrate? These carbohydrates actually act as cellular marker. Okay.
Okay,
For
sir.
example, like say our cell carrying like this kinds of carbohydrates. Okay. So bacterial cell may carrying this like carbohydrates. Okay.
Yes, sir.
So then our immune system can recognize, okay, this cell is ours and this one is a foreign cell. Okay,
Okay.
is it clear?
Yes, sir.
Sir, can you please repeat again?
Yeah, sure. So what is the function of these carbohydrates? These carbohydrates are actually act as markers, okay, in conjugation with the protein or lipids. For example, our cell may carry carbohydrates like this, and another organism may carry carbohydrates like this one, okay. So we can differentiate, okay, this one is human cell and this one is another cell. Is it clear?
Yes, sir.
And if a cells like enter into our system carrying these kinds of marker, then our immune system may detect it. Okay, this is not our cell; it is a foreign cell. Is it clear?
Yes, sir.
Any confusion?
No, sir.
Okay, thank you. Okay, then what is the function of glycocalyx together? Glycocalyx actually helps cell-cell interactions. Okay, and protect the cell membrane from physical forces or stress. Is it
Sir,
clear?
could you explain this one a little?
Which one? Like cell-cell interaction?
Yeah, and the fact that physical action of physical forces and stresses.
Okay, okay, got it. So this is just a layer of one cell. Okay, so here there may be another cell. Okay, so another lipid bilayer. So this glycocalyx may helps to interact with each other. Okay, is it clear? These are actually like proteins or lipid, right?
Yes, sir.
Okay, so this glycocalyx or may interact with another lipid bilayer. Okay, so that's
Sir,
how they may maintain cell-cell interaction. Is it clear?
yes, sir. Sir, that mean can we say that the glycocalyx has like a property of adhesive forces, like when they can interact with another this one? Can we say that they have adhesive forces?
Okay, actually, we know that cells are like compact, right? Our cells.
Yes, sir. Yes,
So
sir.
our body consists of millions of cells. So actually, these cells are compacting together, right? With
Yes, sir.
and they are connecting with one another. So
Yes.
glycocalyx may act as like a connecting device. Okay,
Understood, sir. Yes, sir.
and also when you put stress, okay, so these are actually like breathing with another. So when you put stress, like the stress will divide into everything, right? That are joined together. Okay. So if they are not acting as a connecting device, then the stress might fall into just one cell. Is it clear?
Yes, sir.
And they itself also can take some stress. Okay.
Sir, someone messaged that there are few students who are trying to join in.
Okay, join in. Okay. Any more? Okay, so is it clear? We'll discuss in detail, okay? No problem.
Yes, sir.
Okay, so we have seen two types of protein, right? Integral protein and peripheral protein, isn't it?
Yes, sir.
So can anyone describe about integral proteins?
Integral proteins are mostly embedded through both the layers of the bilayers, and as over here it is written that they have a range of important functions, including like channeling or acting as transporting molecules across the membrane, and some also acts as cell receptors.
Okay, this point I didn't discuss, right? Cell receptor one.
Yeah, so
Okay, so here there might have some types of shape, okay, special shape. So any signaling molecule may comes and bind here, okay? Is it clear?
yes, sir.
So act as receptor means it will accept another molecule, okay, another signaling molecule that can connect with it.
Understood, sir.
Okay, we'll discuss in detail, no problem. Okay, so next, the peripheral protein. Can anyone
Sir, I have
discuss
a
about it?
question.
Yeah, sure.
Sir, the integral proteins are all integral proteins channeling the molecules or only the protein channels?
Oh, not all integral proteins. There are some channel proteins, there are enzyme proteins, okay, receptor protein. Is it clear?
Okay, so they're all doing different things and
Yeah,
transporting
yeah,
different things.
all doing different things, and they are combinedly called integral protein, okay?
Okay, thank you, sir.
Thank you. So, can anyone tell me about the peripheral proteins now?
Peripheral protein mainly situated in the one layer, and it has no channel. That is has the integral protein.
Okay, great.
That's it, sir.
Okay. So, peripheral protein can like detach and reattach with the cell membrane, okay, based on specific signals. And they can coordinate and communicate using network of proteins. So, like they can move from here to another part, okay, and again can join back here. Is it clear?
Sir, then what are the functions of the peripheral protein?
Okay, the main function is like they can transport the signal, okay, and act as signaling molecule. For
Okay,
example,
sir.
this one may act as an enzyme, okay, so
Okay.
that may convert a signal.
Understood, sir.
I know some of you have confusion, but we'll discuss in detail, okay? So here you can see the phospholipids, right? The phospholipids, head and tail, okay? So if we looked at the composition of the head group, what it is made of? Glycerol, phosphate, right?
Yes, sir.
Okay, so here is a structure of glycerol. Okay, how glycerol look like. And with glycerol we can see that there is a phosphate group. Okay, and with phosphate group there may have R group. Okay, so what does this R mean?
An
This
extra
R group
amine
may
group?
be either ethanolamine, choline, anything. Okay, is it clear?
Yes, sir.
And with the glycerol we can see fatty acids are connected. Okay, so fatty acids may be saturated or unsaturated. Okay, so saturated fatty acid means there is no double bond. Is it clear? Saturated fatty acid means there is no double bond. Then what is unsaturated fatty acid?
It has double bonds.
Okay, so it may have like one or more double bonds. Okay, and from the figure we can see that saturated fatty acids are straight, right?
Yes, sir.
And unsaturated, like when there is a double bond, the structure bit curved. Okay, is it clear?
Yes, sir. Sir, are they bent because of the double bond present, which breaks
Yes,
easily because of that?
they bend because of this double bond. Okay,
Oh, okay.
so here may have either like two saturated fatty acid or two unsaturated fatty acid, or one saturated, one unsaturated fatty acid. Is it clear?
Yes, sir.
Okay, can anyone like tell me about the head group now? What the head group carries?
Sir, the saturated and unsaturated diagram on the right corner.
Yes.
Sir, about the carbon and oxygen bonds on top of it, do they belong to the glycerol?
These two?
Yeah,
No, they don't belong to glycerol.
it's part of fatty acid.
It's part of fatty acid and they are connected with glycerol, okay, through bonding.
Okay, so sir, it might be a stupid question, but about the saturated fatty acid not having any double bonds. What about
Yes.
the carbon oxygen bond, the double bond over there?
Where? Here?
Yeah.
Oh, okay. So this is the bond of like COOH group, okay?
Okay,
Acidic
okay.
COOH, right?
Oh, I get it now. Okay, the acidic group. Yeah.
Yeah, acidic group here. Is it clear to you now?
Yes, sir. Okay.
Okay, and here a bonding occurs between this one with this, okay?
Yes, sir.
So here you can see the bond. So can anyone tell me the composition of the head group now? What the head group carries?
The head group carries a phosphate group attached with an R group, while the R group can be an ethanolamine or choline, also combined attached with glycerol. So they're responsible for um, attracting water, attaching to like water particles.
And the tail group?
group mainly consists of saturated and unsaturated fatty acids who are responsible, who I mean, who fear water, repels water, and the saturated fatty acids consist of carbon-to-carbon single bonds, because of which they are mainly straight. But unsaturated fatty acids have carbon-to-carbon double bonds, because of which they are bent.
Okay. Is it clear to all?
Yes, sir.
Sir, I had one question regarding the
Yeah,
R
sure.
groups. Sir, are the R groups like acting as globular protein over here or something?
Globular protein.
Yes,
Oh,
sir.
no. These are actually lipids as well.
Okay. Okay.
Okay.
Okay, sir.
Okay, so what is the role of this fatty acid?
Determining the properties of the membrane and
Okay.
determining how fluid it is.
Okay. So is there any relation between fluidity and saturated and unsaturated fatty acid?
Sir, it could be, yeah.
Okay. So we all know that the plasma membrane contains fatty acid, right? And fatty acid is one of the factors. Okay. So the first question will come then, what is fluidity, isn't it?
Sir, could you repeat again? You were buffering.
So fatty acids are one of the crucial factors that determine membrane fluidity. Okay.
Sir.
So my point is, like, what is membrane fluidity?
Membrane fluidity is maybe like how much the membrane can move, and because of which, like, how much substances or things it can let inside it or not. Like the permeability depends on the fluidity, the fluidity.
Okay, permeability, yeah, it's linked, but okay.
Sir, maybe I think that fluidity will consist of water. So it will determine the hydrophilic side and the hydrophobic side of the cell membrane, maybe.
Okay, so in fluid, we can see like fluid can move, right?
Yes, sir.
Sir.
So in plasma membrane, like these molecules can move around, okay? They can float. For example, like this one may float from here to here, okay? Is it clear?
Sir.
And these bilayers also can move, like from here, this one can move to here, okay? So this is
Sir,
fluidity.
sir, if the fluidity is more, will it be advantage for the cell, or like is it a disadvantage for the cell?
There must have to be a balance, okay?
Okay.
Too much fluidity is not welcoming, and similarly, like if it is too rigid, then it's bad as well, okay?
Okay. Okay, sir.
Okay, so there are two types of fatty acids, right? Saturated and unsaturated fatty acids. So saturated fatty acids are mainly more compact, okay, and tightly packed. From the structure, you can see that they are straight line, right?
Yes, sir.
Yes, sir.
So if you see if there is another fatty acid here like this, just a single straight line, okay? And then it looks like more compact compared to unsaturated fatty acid, right?
Yes, sir.
Okay. So if the temperature is cool, then saturated fatty acid pack together very tightly, okay, and make the cell membrane dense and rigid, okay. So rigid means like they're tight, okay. Is it clear?
Yes, sir.
And unsaturated fatty acids are like in band form, right?
Yes, sir.
Sir.
So this band form actually helps to maintain the fluidity, okay. So if there is like more unsaturated fatty acid, then the membrane will become more fluid. Is it clear?
Yes, sir.
Yes, sir.
So at lower temperature, if the membrane becomes so rigid, then it might break down, okay. So if it carries like extra unsaturated fatty acid, then it will not break. Is it clear?
Yes, sir. So that means that there should be a proper balance of unsaturated fatty acids and the saturated fatty acids as well.
Yes, yes.
Okay. So, sir.
Is it clear to all?
Yes, sir.
Okay. And what will happen to high temperature then?
The saturated fatty acids won't be packed tightly together. They won't be as compacted.
There's a
Or that's
lipids,
what I
right?
think.
So if you hit the lipids, they will move, right?
Maybe.
Like if we hit the oil,
Oh, yes.
yeah, their level
Yeah.
increase, right?
Yeah.
So with the rise of temperature, the fluidity of the membrane will increase, isn't it?
Yes, sir.
Yes. Yeah.
And if it carries like too much unsaturated fatty acids, then they may like detach from each other, okay?
Yes, sir.
Is it clear?
Yes, sir.
Okay, then at too high temperature, saturated fatty acid may be beneficial, isn't it?
Yes, sir. Sir, is this temperature the inner temperature of the cells or the temperature of the environment?
This is just hypothetical, just if the temperature rises, like our body temperature is 37, right?
Yeah.
So if we come in contact with like high temperature, say like 40 degrees centigrade, so the membrane fluidity will increase.
Oh, okay.
Okay. So actually our body temperature is very tightly regulated. So like whether the surrounding temperature rise or fall, our body always maintain 37 degree. Okay. And if some reason like body temperature decrease, then we call hypothermia. Okay. And if the body
Yes, sir.
temperature increase, then we call hyperthermia. Okay, so we have seen another component that we didn't discuss that much, that is cholesterol, right? Are you hearing me?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, so cholesterol is another factor that maintain plasma membrane fluidity. Okay, and cholesterol only present in animal cells. Is it clear?
Yes, sir.
Okay, so at lower temperature, how cholesterol may help?
Sir, cholesterol only remains where? I missed that one, sorry.
Okay,
Makes sense.
so here you can see the cholesterol clearly, right? This is a cholesterol.
So in between the phospholipids only.
Yes, just one.
Okay.
Okay, so cholesterol only present in animal cells. Okay. And also it changes fluidity with temperature. Okay.
Yes, sir.
So at lower temperature, cholesterol increase the fluidity. And at higher temperature, cholesterol reduce the fluidity. Is it clear?
Yes, sir.
Okay. So without cholesterol, if the temperature decreased, then the membrane will be rigid. Okay. And the fluidity will decrease and the membrane may break down. Is it clear?
Yes, sir.
Yes, sir.
And if the temperature is so high, then the membrane will separate each other. Okay, separate from each other. Is it clear? So they will
Yes, sir.
not be in right shape.
Yes, sir.
So cholesterol present only in animal cells. Then what happened to plant cells or like bacterial cells? Any idea?
Um, I think it's vague, but they have cell walls, so that might help with their fluidity or something. Might be wrong about this.
Okay, any idea? Any more? Anyone?
Sir, what he said that they have cell walls, so they don't really
Tree
need
has,
cholesterol
sir, might
to
be tree
maintain.
has own factor like gum.
Okay, own factor. Okay.
Sir, they die at high temperature, I'll say that.
Okay. So actually, plant cell contains phytosterol. Okay. So they are similar like cholesterol. Okay. So why the term is phyto? Phyto means plant. Okay. Is it clear?
Sir.
And they have same activities like cholesterol. Okay.
Sir, so their functions are similar to what cholesterol functions are?
Yeah, similar, almost similar function.
Okay, sir.
Okay. And bacteria contain haploids. So haploids maintain the fluidity in bacterial cells. Is it clear?
Yes, sir. Sir, should we memorize or remember the skeletal structures of organoid, phytosterols, cholesterol, and all of those? Should we learn
Uh,
them?
if you wish, you can learn, but I think for this topic, you don't have to memorize this. Okay.
Okay, alhamdulillah.
So we have like another topics like carbohydrates, lipids, proteins. There you can memorize. Okay, the structures.
Okay, sir.
Is it clear?
Yes, sir.
So I don't want to like make this lesson more complicated. Okay.
Yes, sir.
Okay. Thank you. Okay, so there is a model named fluid mosaic model. I think everybody of you heard it, right?
Yes, sir.
Especially in high
Yes,
school
sir.
level. So can anyone tell me about this model?
Sir, protein and lipids are together formed in such a way that looks like a mosaic. So that's how the mosaic model name came actually, I think.
Okay, so who discovered this model?
Singer and Nicholson.
Okay, great. So actually we already discussed about this model, okay? So this model is for plasma membrane, right?
Yes, sir.
Yes, sir.
Okay, so we all know that plasma membrane contains proteins and lipids, isn't it? And lipids bilayer has like dual role. They can act as solvent for membrane protein, and also they act as like permeability barrier. Okay. So here the structure of plasma membrane, or cell membrane. So why it is called fluid mosaic? The point is here. Okay. So the floating issue I have discussed, right?
Yes, sir.
Yes, sir.
Can anyone tell me the floating issue?
There are different molecules that float along the lipids through the cell
Okay, great.
membrane.
Thank you very much. And why it is called mosaic?
Because it has a mosaic-like appearance from the top view.
Yes. Is it clear to all?
Yes, sir.
Okay, so another topic, membrane diffusion. Okay. So what does membrane diffusion means? If I simply say membrane can diffuse, okay. So there are two types of diffusion, lateral diffusion and flip-flop diffusion, okay. Is it clear?
Yes, sir.
Okay. So lateral diffusion, like what happened in lateral diffusion? One can move to another side, okay. Is it clear?
Yes, sir.
Sir, could you please explain that again?
What happened in lateral diffusion? One can move to another side, okay.
Okay.
So this is phospholipid, right? Head group fatty acids, right?
Yes. Yes, sir.
Yes,
Okay.
sir.
So one can move from this side to here or this side to here like this. Is it clear? And this process is very rapid, okay. And another diffusion may possible, that is flip-flop. So this one may move to here and this one will move to here, okay.
Yes, sir. Sir, I have a question based on this.
Yes, sure.
Sir, why do membranes need to diffuse from one position to another position? What is the main reasons to do it?
Okay. So you may put stress, okay, on the membrane. So like you may put pressure here, so it will move from here, right?
Yes, sir.
And like after certain point that you will take the stress out, okay. So it may be like moves from here to another direction, and this one may like come to the top, and this one may replace again. Okay.
Okay. Okay, sir.
And
Understood.
membrane is like fluid. Okay, so they are
Yeah.
actually moving.
They're always moving.
Yeah, they're always moving.
Okay. Okay. Understood.
So
sir.
this is possible to like it can replace another position.
Understood. sir.
So what is the function of cell membrane? I feel this one will be more clear. You are seeing the screen, right?
Yes,
Yes,
sir.
sir.
So
Yes,
the
sir.
main function is membrane act as boundaries. Okay. Second, it localize some organelles like mitochondria, chloroplast, nucleus, and it can transport molecules. Okay, like nutrient molecule. It can detect signals. It also communicate with another cell through cell-cell communication. Okay, and it join with another cell through cell-cell adhesion. Is it clear?
Yes, sir.
Okay, now we'll discuss the major functions of membrane proteins. Okay, so how the transportation occurs? So we discussed that plasma membrane contains channels, right?
Yes, sir.
Okay, so hydrophobic molecules that may pass through these channels. Okay, is it clear?
Yes, sir.
And if we need to like move out anything from the cell, then we can also use the channels. Okay, and consider here that you are moving out water through a motor. Okay, so when we turn on the motor to move water, then we need current, right?
Yes, sir.
So here the cell also use current like energy source. Okay, so what is energy
Yes, sir.
source here?
ATP.
Okay,
Sir, ATP.
is it clear to all?
Yes, sir.
Yes, sir.
Sir, during the hydrophobic, we don't need energy, like when hydrophobic molecules move inside.
It may need or may not.
Okay, sir.
And when you move
So, sir,
something out of the cells, it may need energy or may
yes, sir.
not. It's just a
Okay, sir.
representation. Okay.
Okay, sir. Understood.
Okay, and this protein may also act as enzyme. Okay. So we all know about the activities of enzyme, isn't it?
Yes, sir.
So what is the function of enzyme?
They either break up a molecule or attach a molecule. Like they either go under catabolism or anabolism that like usually like normally speeds up the reaction without getting used up.
Okay, if I simply say they catalyze a chemical reaction. Okay.
Yes, sir.
And this membrane protein also helps to transduce signal. Okay.
Okay, sir.
So there may have receptor like this, and signaling molecule may bind with this receptor. Okay. So when signaling molecule binds, they pass this signal inside the cell. Okay. Is it clear?
Yes, sir.
Is it clear to all?
Yes, sir.
Here, hormone may act as a signaling molecule. Okay. So when signaling molecule binds, the conformation or the structure of this protein may change a bit. Okay. Is it clear?
Yes, sir.
Okay, so this is a glycoprotein. Okay. So glycoprotein actually consists of carbohydrates and protein, right?
Yes, sir.
Yes, sir.
So as we discussed earlier, this may act as a tag of cell recognition. Is it clear?
Yes, sir.
Yes, sir.
Okay. And also this protein may act as intercellular joining. They may help in intercellular joining. Okay.
Yes, sir.
And also they may attach with the cytoskeleton or also extracellular matrix. Is it clear?
Yes, sir.
So can anyone discuss about the transport function now? Just in very short.
There are protein channels present in the um, so in the phospholipid bilayers or in the plasma membrane where um, the particles can get inside without the ATP energy also, or and it's selective particles, not every kind of particles. Selective particles can get in through the with the help of energy or without the help of energy. Same way, particles can also get out of the cell by using energy from ATP or may not using energy from ATP, depending on the situation.
Okay. So here one point, this is termed concentration gradient. Okay. So when it need ATP, when it don't. Okay. There is a term called concentration gradient. If the concentration of something is higher outside and we need to take this thing outside of the cells, okay, then we are working against the concentration, right?
Yes, sir.
So then we need to use ATP. Okay.
Yes, sir.
Same here as well. If something concentration higher is here and we are taking inside the cell, then we might need ATP. Okay. Is it
Yes,
clear?
sir.
Okay. Can anyone describe about the enzymatic activity now? Just in very short. Anyone? Is it difficult?
Sir, I understood, but it's quite difficult to explain it.
Okay. So you try just your own way. Okay, no problem.
So enzyme catalyzes the metabolic activities in the cell.
So enzymes actually act as catalyst, right?
Yes.
Yes.
They try to help in chemical reaction. Okay. Is it clear?
Yes, sir.
So in our second lecture, I guess, we discussed about an enzyme, right? Second or third lecture. What is the name of this enzyme? Okay, you can check. Hexokinase, right?
Yes, sir.
Okay, so
Yes, sir.
enzyme just help in conversion of one molecules into another form. Okay. So this membrane protein may act as enzyme. Is it clear?
Yes,
sir.
sir.
Okay, and membrane protein also helps to pass chemical signals. Okay, so how they can pass chemical signals? When signaling molecules binds with the receptor. Okay, is it clear?
Yes, sir.
So this is a receptor protein. So when the signaling molecule binds with it, it passes this message below, okay, inside the cell. Is it clear?
Yes, sir.
And this three is very clear to you, I feel, isn't it? Cell recognition, first of all.
Yes, sir.
Yes, sir.
Can anyone tell me like how cell recognition happen?
There are tags to the tags. Acha, you can explain, no problem.
No, it's okay. Go on.
There are glycoproteins that act as identification tags to specify or help the other cells to get recognized.
Okay. And how intercellular joining happen?
Sorry, sir, I didn't understand what.
How intercellular joining happen like this? The joining. So one membrane protein
Is
may
it
hook
by
with
active site?
uh, no, here is nothing active site. Nothing about active site here. So one membrane protein may hook with another membrane protein. Okay. So hook means like it might join or interact with another membrane protein. Is it clear?
Yes, sir.
Okay. The last one. Can anyone tell me the last function?
Basically, the cytoskeleton or even the microfilament tubes, they can get attached to the proteins, which usually helps with maintaining the cell shape and uh, like stabilizing the position of the specific membrane proteins in it. Then it also uh, attaches, or you can say adhere to the extracellular matrices which are outside of the cell, and usually this helps with coordinating whatever is extracellular, or even if there is any intracellular changes inside the cell.
Okay, thank you very much. So our last topic, cell wall of bacteria. Okay. I know previous two slides were like a bit advanced for you, so I have organized a very easy slide for you now. Okay. And I didn't like, I have no plan to discuss in details about the cell walls, okay, because you will learn this in other courses. Is it clear? So we'll learn just the basic things. So there are two types of bacteria we know, gram-positive bacteria and gram-negative bacteria, okay. So their cell wall is different, okay. So how they are different? They're different in terms of outer membrane, okay. Is it clear?
Yes, sir.
And in terms of peptidoglycan layer. So in gram-positive bacteria, there is no outer membrane, okay. Here you can see the outer membrane. And gram-positive bacteria contain a dense layer of peptidoglycan, okay. Is it clear?
Yes, sir.
Yes, sir.
Okay, and in the outer membrane, there is lipopolysaccharides, okay. So saccharides means carbohydrates, right?
Yes, sir.
So these are actually glycolipids, isn't it?
Yes, sir.
Okay. So there's membrane also have like different functions, like in terms of infectivity and sensitivity to the, like drugs, okay. So which bacteria might be more sensitive to drugs? This one or this one?
Gram positive, sir.
The gram-positive.
Sensitive, right?
Yes, sir.
Why?
Because
It
mainly because
lacks
of the
outer
outer
membrane.
membrane.
Okay. Then the drug can easily pass through it, right?
Yes, sir.
Okay, here it's a bit difficult to pass through, isn't it?
Yes, sir.
Yes, sir.
Okay, so what are the basic differences between the gram-positive and gram-negative bacteria? If I asked you to select one, peptidoglycan layer, right?
Yes, sir.
Okay. And how can you differentiate between gram-positive bacteria and gram-negative bacteria in the lab? Is there any procedure?
Yeah, the gram staining procedure.
Okay. So if we do the gram staining procedure, then we can differentiate gram-positive and gram-negative bacteria. Then how can we differentiate? Based on the
Color.
color.
Purple
Okay,
colored
great.
stain.
Sir, the color mainly shows because of the outer membrane, right? Because of how it reacts with the outer membrane.
The peptidoglycan layer.
Oh, okay. Understood.
Okay, so the gram-positive bacteria retain the crystal violet color. Okay. Is it clear?
Yes, sir.
Yes, sir.
and gram-negative bacteria doesn't. So it's look like purple if you see in the microscope, and it's look like reddish. Is it clear?
Yes, sir.
Any question from your end?
Sir, question was there in the last slide. Little.
This one?
Yeah, the last slide, sir. Not this one. Yeah, this one, sir. Sir, what is this porine over here? In
Okay,
the
porine is a
outline?
protein. Okay.
Okay, sir. Could you give us like maybe little brief explanation about it? Just to like
Okay.
have a general idea of what it would be.
Okay. So porine protein actually like control the molecule passing through the bacterial membrane. Okay.
Okay, sir.
Passing
Yes, sir.
through, passing out. Okay. Any question?
No, sir.
Have you completed the assignment,
Yes, sir.
or you are still doing?
Not yet, sir. Still
Sir,
doing.
still doing.
We're working
Oh,
on it.
okay.
I have submitted it to you.
Okay. Thank you.
Sir, you don't have a Slack channel yet, do you?
No, till now not, because I'm just waiting for the Brachio ID, and I feel that it may come like within 2 or 3 days, but it's just waiting me. Okay. So
Sir, in
in that
bug,
case, we can go for a solution, right? So I don't know like how long it might take. So I feel
maybe
that we
like
need to
messenger
create a
group.
group, right?
Yes, sir.
So where we can create a group easily?
Maybe on WhatsApp.
No, I don't use
Messenger.
WhatsApp regularly, actually. It could be a
Uh,
Facebook group or a messenger.
Messenger, sir.
Sir, you can also open a Google Classroom.
yeah, I had opened a Google Classroom just for a trial, okay, for BCS 102 course, and it doesn't work with your G Suite ID, okay?
So, for example, if I face any complications or if I want to ask any questions while studying through your lectures, do I just mail it to you about the questions?
Yeah, you can surely email me.
Okay, sir.
Sir, I guess
So
for
regarding
now,
the group,
a
what we
messenger
can
group
do,
would be good, I guess.
is there any suggestion from your end?
Sir, maybe a messenger group will be convenient right now, as most of them are saying messenger is convenient for them.
Okay, messenger group. What about if we create just a Facebook group?
Yeah, so that works too.
Okay. Okay, then we'll create a Facebook group, okay? And then we will join one by one.
Sir,
Is it clear?
are you going to open the Facebook group?
Because yeah, it's taking long time, so you need a group. So what I can do
Yes,
for
sir,
you
we
now?
need
Yeah.
enough communication. Yeah. So, sir, if
Sir,
you create a Facebook group and then we all get added, so maybe we can type our concerns and everything, and maybe you could respond to that, and maybe we could respond to you too.
Yeah, sure. Thank you very much.
sir, in bugs, recently I saw that there was a BCH course open.
Okay,
Did
so
you open it, sir?
no. I'll let you know when I open it. Okay.
Okay.
So there is like other section, right? There is
Yes,
like
sir.
mine one is section 3, so section 1, section 2. Okay.
Understood, sir.
Okay, so I'd like to take the attendance now. So serial number 1. 2, 3,
Yes, sir.
4,
Present, sir.
5,
Present, sir.
6,
Present, sir.
7,
Present, sir.
8, 9,
Present, sir.
10, 11,
Present, sir.
12,
Present, sir.
13,
Present, sir.
14,
Present, sir.
15,
Present, sir.
16, 17,
Present, sir.
18,
Present, sir.
19,
Present, sir.
20, 21, 22,
Yes, sir.
23, 24,
Yes, sir.
25,
Present, sir.
26,
Present, sir.
27,
Present, sir.
28, 29, 30,
Present, sir. 29
20.
present.
Oh, okay, thank you. 30, 31,
Present, sir.
32,
Present, sir.
33, 34,
Present, sir.
35, 35, 36,
Present, sir.
37,
Present, sir.
38,
Present, sir.
39,
Present, sir.
40, 41,
Present, sir.
42.
Sir,
Yes.
sir, Miskatul Jannat and Zarin Tasneem, both of them are present, but their mics are not working.
Okay, okay, no problem. I'll sort it out.
Okay, sir.
43,
Present, sir.
okay, serial number 1 and 2, right?
Yes, sir, I guess.
Any question?
Sir, did you give me as present serial number 35?
Serial number 35, okay. No.
My mic wasn't working.
Okay,
Sorry.
35 present, okay. Anyone?
So did you assign serial number 26 as present because he has a problem with his microphone?
27,
26.
26 present, yes.
Okay, thank you, sir.
Okay. Thank you very much for joining in the class. Bye for today. See you in the next class. Stay safe. Allah Hafiz.
Assalamu alaikum,
Thank
sir.
you,
Allah
sir.
Hafiz.
Bye, bye, sir.
Bye, sir.
Assalamualaikum, sir.
Allah Hafiz.